I'm Janet Rossant, and I'm President and Scientific Director of the Gairdner Foundation. I'm excited to be with you today to announce the recipients of the 2024 Canada Gairdner Awards, given to scientists whose outstanding research contributions are helping us understand human biology and human health with impact around the world. Before we begin, I wish to acknowledge the land on which Gairdner Foundation operates in Toronto, Canada. For thousands of years, it's been the traditional land of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and continues to be home to many diverse First Nations, Inuit, and Métis peoples from across Turtle Island. We are grateful to work together on this land. The Gairdner Foundation began as the vision of Canadian philanthropist Jim Gairdner in 1957 to recognize and reward researchers around the world whose work has changed our understanding of biology and human health. The awards have grown in international prestige over the years, and with further investment from the Government of Canada, continue to be an iconic emblem of Canada's position on the world stage. For over more than 6 decades, the Gairdner Foundation has awarded 418 scientists from more than 40 countries for their incredible work, 98 of whom have gone on to win a Nobel Prize. Annually, 5 Canada Gairdner International Awards are given for biomedical research, along with the John Dirks Canada Gairdner Global Health Award, which is given for impact on global health issues. And last year, we introduced an entirely new award, the Canada Gairdner Momentum Award. 2 Momentum Awards are reserved for mid-career Canadian researchers with exceptional scientific contributions that impact human health. I encourage you all to please share the news on social media using #Gairdner Awards and tagging us at Gairdner Awards. We pride ourselves in the caliber of our laureates, who are selected through a rigorous multi-level adjudication process, which involved leading scientists from all corners of the globe. I'd like to thank the members of our Medical Review Panel, the Medical Advisory Board, the Global Health and the Momentum Award adjudication committees for their hard work in selecting this year's outstanding cohort. I'd also like to take a moment to thank our partners. Gairdner is supported nationally by the Government of Canada and the Canadian Institutes for Health Research. Their partnership is essential for bringing our laureates to Canada with associated programming across the country, providing students, trainees, faculty members, and the public access to the groundbreaking science that is changing the world. The Gairdner Foundation is also supported by our media partner, the Globe and Mail, who help us share the work of our laureates with the public. And now, without further ado, it's time to announce our 2024 laureates, beginning with the Canada Gairdner International Awards. These awards recognize outstanding biomedical scientists whose seminal discoveries have significantly increased our understanding of human biology and disease. The first 2024 Canada Gairdner International Award laureates are Dr. Shankar Balasubramanian, David Klenneman, and Pascal Meyer. Together, they are awarded for their fundamental and applied research that led to a revolutionary and affordable method to sequence DNA on a massive scale, so-called next-gen sequencing, which has dramatically accelerated discoveries in the life sciences and breakthroughs in identifying the genetic underpinnings of disease.
I'm Pascal Meyer. I'm a biophysicist from University of Strasbourg. So I was involved during my career in what I call DNA analysis technologies. The problem that was back in '96 is that with the existing DNA sequencing technology, each time when you wanted to read a bit of DNA, you needed to have one vial, one sample, one operation. And so for sequencing the whole human genome, you had millions of such operations to do. So that made it very expensive, very cumbersome, very long. So my idea was to have something where you could use the whole DNA genome, cut it in pieces, and put it on any surface in such a way you can eventually assay them. This is very difficult to do on one single DNA fragment. And we were progressing in getting the technology together. We knew 2 other companies working on it, which were Lynx Therapeutics and Solexa with Shankar and David.
My name is David Klenneman and I'm a professor at the University of Cambridge.
I'm Shankar Balasubramanian. I'm a chemist at the University of Cambridge.
The Human Genome Project was finishing, which was the first sequence of a whole human, but that was the average sequence of a human. And what was required was a technology that could sequence individuals. So that's 3 billion bases. And we realized we could do that by making the whole method massively parallelized and miniaturized.
And today this technology is able to sequence DNA more than 1 million times faster and lower cost than at the start of the project. David is a physical chemist and he works with lasers and imaging. My background is more organic chemistry, biochemistry, and so we pushed each other's ideas along. And I think mobilizing the resources, the people, the expertise, and then solving all the individual problems and bringing them together and into an integrated system, I think was a considerable challenge.
So the technology originally started off as a research tool, but it's been fantastic that it's now being used widely in hospitals actually to identify what's wrong with people and then find cures in a lot of cases. So that's very gratifying.
So to me, the Gairdner Prize is a very strong academic recognition of the work that has been done. And so this is very important because it gives much stronger and scientific kind of recognition, which is something I'm very happy to have, of course.
I think the award just shows you that how powerful science is and how by doing basic science and doing curiosity-driven science, you know, we can go from an idea literally written down on a piece of paper to 20 years later, technology that's used all over the world and in hospitals and starting to save people's lives. And I think the importance of the award, particularly for the young people, is to show what's possible, where scientists working together can solve major problems and make a big difference.
Congratulations to Drs. Balasubramanian, Klenneman and Meyer. The final Canada Gairdner International Awards of 2024 go to Dr. Zelig Eshar and Michel Sadelain. They are awarded for their seminal contributions to the conception, development, and application of CD19 CAR T-cell therapy for cancer.
My name is Michel Sadelain. I'm a researcher, medical researcher in New York City, where I direct the Center for Cell Engineering at Memorial Sloan Kettering Cancer Center. Several decades ago, we formed the idea that we could perhaps help our immune system by instructing T cells, providing a genetic instruction to tell them what cancer cell to recognize and then to go on to eliminate those cancer cells. And this is what CAR therapy is about. And the CAR molecule that is provided to those T cells now will guide the T cells once they are reinfused into the patient to the tumor site. And upon binding of the modified T cell to the tumor cell, it initiates a program in those T cells that should lead to the destruction of the tumor cells. And today there are 4 CAR therapies approved by the US FDA, also approved in Canada and in Europe and other countries, that are available to patients with refractory B-cell malignancies. But the concept is applicable to a broad range of cancers, and today it's really wonderful to see so many labs around the world seeking to adapt this approach to other cancers.
What I did, actually, I modified these T cells by engineering their receptors so that they will recognize the tumor. So we engineer the cells out of the body, the immune cells, then we put them back into the patients, and this cell will recognize the malignant cells as foreign or as different. And then what they will do what they know. They have the mechanism to reject, eliminate, and this actually has been done. I myself working on it for more than 20 years. So we started it in 1986, working on it with a group of students. When I heard that it really can cure patients, I was happy, the happiest man in the world. However, I knew it should work. And I believe the knowledge that accumulates and the researchers all over the world will contribute to it. And so our small beginning that took so long time to accomplish has a future for the patients. And that's what we do.
The Canada Gairdner International Award is one of the most prestigious science prizes in the world. It's not just a personal recognition, it also signifies to many of us who pursued this approach for many years in the days where there were many who didn't believe that it could ever succeed. It is a sort of a validation of all of these efforts by so many.
Congratulations to Drs. Eshar and Sadalan. Next, I'd like to announce our 2024 John Dirks Canada Gairdner Global Health Award. This award is given to a scientist whose advances have a significant impact on the health and well-being of those facing health inequities worldwide. The 2024 award goes to Dr. Gagandeep Kang. She is awarded for her extensive cohort-based epidemiological, environmental, and clinical trial research on enteric diseases in children and their effects on life course, with significant impact on vaccine development and health policy in India and internationally.
I'm Gagandeep Kang, and I am a public health virologist. I've worked in public health in India pretty much my whole professional career. I started out studying something very unglamorous, diarrhea, and I've stuck with it my whole life. If we think about all of the problems that people have that are related to health, essentially respiratory diseases and diarrhea are the biggest killers of children in poor countries around the world. And what we wanted to do was to figure out does it take to make a vaccine that will protect children from diarrhea due to rotavirus. We have been able to make in India 2 vaccines that are now being used to immunize about 3/4 of the children that live in the poorest countries in the world. The team that I've been working with, I started out with 1 technician and 1 freezer in 2000 when I came back from postdoctoral training in the US. The first technician that I started with, Sheila, still continues to work with me. The Gairdner Award is a recognition of contributions to global health, and to me it means that the work that my team and I have done over a really long period of time is something that matters for public health, that we have been able to deliver work of very high quality that has made a difference to children in India and around the world. To me, this is particularly important because all of the work that we did was designed, led, done in India, and has led to interventions from India reaching the world. We've gotten to a stage where the bulk of vaccines for childhood immunization come from India. That's really local for global.
Congratulations to Dr. Kang. Finally, I'm delighted to announce the 2024 Canada Gairdner Momentum Award laureates. These awards are given to mid-career investigators in Canada who in their last 6 active years have produced exceptional scientific research contributions with continued potential for impact on human health. The first 2024 Canada Gairdner Momentum Award laureate is Dr. Megan Azad. She is awarded for research on understanding how human breast milk contributes to shaping the infant microbiome and lifelong health.
My name is Megan Azad. I'm a professor of pediatrics and child health at the University of Manitoba and a research scientist at the Children's Hospital Research Institute of Manitoba. And I co-direct the Manitoba Interdisciplinary Lactation Center, or MILK with a C for short. Well, my team studies breast milk and breastfeeding and early nutrition and development of child and infant health. We're interested in understanding what's in breast milk, and that's not a simple question. There are, of course, nutrients, but there is also so much more than that. So hormones, antibodies, microbes, immune cells, and we're really trying to understand that from a systems perspective. So how do all those things work together? And then how does that influence infant health? Initially, my research focused on Canadian families, and in that study, we had about 3,500 mothers, babies, families participating. And then lately, I've been moving more into international work, so we're starting to look at how is milk similar and how is it different in different settings around the world, and how is it affecting babies in those different settings. We look at, honestly, hundreds of components in the milk because there are just so many things to look at. And what we have found is that there is what we call a dose response. So the more breast milk, whether it's more exclusive or longer duration, the better the health outcomes for these babies. So we see lower rates of asthma, lower rates of obesity. And I think what's a nice message for parents is that kind of every drop counts. So I think there's a lot to learn there, both about just what's in breast milk. Honestly, I think we'll discover some new components that haven't been described yet, and also information about how they interact. And then we can use that for understanding how breast milk works, and there's many ways we could apply that knowledge. And that's both in our own setting here in Canada for conditions like food allergies and obesity, which are increasingly prevalent, but also in these other settings we're working in where the problems are different, wasting and stunting of babies. And so what can we do in all of those scenarios and learn from human milk? Winning a Gairdner is really special. It's a huge honor, and it recognizes the work that I've done together with my team. I think recognizing this field of work also is special to me because I think historically women's health broadly and lactation and breastfeeding specifically has really not been adequately researched, adequately funded, adequately taught in medical school. So to have an award of this caliber recognize research in this area, I think does a lot for the field as well.
Congratulations to Dr. Azad. The second 2024 Canada Gairdner Momentum Award is given to Dr. Christian Landry. He is awarded for the development of novel approaches that combine synthetic biology, experimental evolution, and systems biology to address fundamental questions about gene function relevant to health and human disease.
My name is Christian Landry. I'm a professor of biology and biochemistry at Université Laval in Quebec City. So in my research group, we study mutations, and mutations are changes in DNA that occur spontaneously. Mutations are what causes many human diseases. It is what causes cancer in many cases. And one context in which mutations are super important is that they cause sometimes resistance to drugs. So what we like to do in the lab is to use tools of synthetic biology so that we can create many, if not all, the possible mutations and study their impact on drug resistance. Winning the Canada Gairdner Momentum Award means a lot because it means that people outside of my specific field are recognizing the work that we do as being important for knowledge in general, but also for biomedical research specifically. Let's say first that every day we get a little bit of satisfaction in science. We discover things, but what we discover with time, especially when we go back home and we talk to people outside of science, it doesn't translate into things that are super important for other people because progress is very slow and they're looking for things that happen very fast. You would like at some point that people outside also appreciate what we do. And this is what happens with awards like this one. Coming from outside, it's particularly more rewarding, I think. So I come from a family where there's no one went to university. What people cared about when I was a boy in high school was that boys would finish high school. So I wasn't clear in my life that I would become a scientist. So I cannot say that I was supported to become a scientist. I was supported to do what I wanted to do, which is, I think, probably even more important because I feel that I chose what I wanted to do. I met my wife when I was a master's student, and now she's my colleague. So we went through all the steps of a scientific career, starting a lab and applying for the first grants, getting your papers rejected or accepted. We've been through that together, so it's been super important for me.
Congratulations to Dr. Landry. As president here at the Gairdner Foundation, I'm thrilled to welcome these 8 phenomenal individuals into the Gairdner family a distinguished group of researchers at the helm of many of the world's most significant advances in health-related science. A large part of Gairdner's core mandate is inspiring the next generation of researchers through our Pan-Canadian outreach program. Over the course of 2024, Gairdner will send our laureates and other outstanding scientists across the country to share their science and stories with faculty members, trainees, students, and the public. Please check out gairdner.org for a full listing of all our outreach events. Follow us on social media and subscribe to our newsletter to get updated information on everything Gairdner. Congratulations again to our 2024 laureates, and thank you for joining us today. Now I invite you to join us for a live conversation with some of this year's laureates, where you will have a chance to ask them questions and share your congratulations. Hello, everyone. Here we are live now and ready for conversations with our laureates. I hope you enjoyed the video, which gave you a taste of the kind of research and the background of our amazing laureates that we have again this year. So some housekeeping notes. You have the opportunity to enter in the chat any questions that you would like to address to any of our laureates. We will see what we can do. I have some questions to start with, and if you put questions in the chat, we'll try to get to them. I can't guarantee we'll get to all of them, but they will be monitored and we'll try to get to them as we go. If you have any issues, technical issues, there's a help link on the top right of your screen if you need any help there. So I'm going to bring our laureates to the stage, the virtual stage, and have conversations with them. And we're going to begin with all the Canada Gairdner International laureates. So we're going to bring Dr. Balasubramanian, Dr. Kleinerman, Dr. Meyer, and Dr. Sadlan to the stage. So welcome, everybody, and it's great to see you. Congratulations again to all of you on the award. It really it's a great pleasure for me to actually meet you. Michelle, we've met before, but the others haven't met, so this is our first Zoom meeting. It's very nice, and of course I look forward to meeting all of you in person in Toronto and across the country in October. So I'm going to start with some questions for Doctors Balasubramanian, Kleinerman, and Meyer. We're going to be talking about next-gen sequencing, and I'm going to start with you, Shankar. When you look back at the development of the work that you did in collaboration with Dave, collaborating in the 90s, you were building, as I understand it, you were really looking at single molecules. You were building a fluorescence microscope to study how DNA polymerase works at the single molecule level with fluorescent tag nucleotides. And that's hardcore chemistry, biochemistry. At what point did it occur to the two of you that this could actually be translated up into a process that could be scalable to billions of DNA bases, not single molecules?
Thank you, Janet, and thank you to the Gairdner Foundation. It's an honor to be here. First point I would make is the work that we were doing was very basic, curiosity-led science. We weren't actually trying to do anything technological or even do anything useful. We were just exploring. And what we realized along the way is we tried different setups to carry out the experiment. And then the one we settled on, we realized inadvertently could be used to read the sequence of a strand of DNA in principle. And I'd say the big moment was Dave is a physical chemist, and he was thinking about how to image all this. And we could see a way to image literally millions and millions of different sequences on the surface and actually watch this process of DNA synthesis that could be developed to decode DNA in parallel. And then when we did some back of the envelope type calculations, we saw a way to sequence on a human genome scale, billions of bases. And so that was really the time that we got very excited about this. And of course, being able to sequence on the scale of many individual genomes, we could see a world in the future where you could sequence on a population scale to find out all the genetic causes of many health conditions. And that's the world we're in today, of course.
Right. So yeah, I mean, when you at that point in your time, looking forward to where we are today, did you really have that vision? We're going to be sequencing the population of the world?
Well, the vision was to see a way of sequencing on a population scale. Because of the differences between individuals, that's really the only way to get at the basis of why we're different, why there are susceptibilities to certain diseases, and then also genetic diseases that occur in all of us. So that was the goal, and it's still in play, of course, with the many wonderful scientists and clinicians using the technology, but very, very pleased we've got there.
It's amazing. It's transformational for sure. So I'm going to turn to Dr. Kleineman. So Dave, I know that your current research is really much more on not DNA but protein aggregates in neurodegeneration. Does that build on similar approaches to sort of the basic science that was underpinning the DNA work?
No, exactly. First of all, I'd just like to congratulate all the other winners before I answer your question. But exactly, the same instrumentation that we can use to detect single molecules of DNA, we can use to detect single protein aggregates. So we're looking at the very small protein aggregates that form on the way to forming larger aggregates that ultimately end up as the amyloid plaques and tau tangles that characterize diseases such as Alzheimer's disease. And so we're trying to understand the mechanism, how these aggregates form. And we're particularly very excited that we can actually detect these aggregates in human blood and serum and plasma. And we see changes as people develop the disease. We're hoping this might be a way of developing ways to diagnose people at very early stages of disease, where any therapy might work much more effectively.
That's extremely important and very exciting. So you can win 2 Gairdner Awards. They have to be for different areas. So keep working. Okay, I'm going to now turn to Pascal Meyer. And Pascal, your technology, which was the sort of clustering, was a key component to the sort of the scale up to massive parallel sequencing on a scale that's never been seen before. Again, perhaps the same question that I asked Shankar. When you were developing this, did you imagine we'd be where we are today with the ability to sequence genomes at extreme speed, many genomes, population level analysis? Did you see it back then?
Yes, I did, and congrats also to everyone. It's sometimes it's seen as pretentious, but it was not. Yeah, you were just. I was realizing that if we could make the amplification on the surface, we would get small DNA colonies of the size of a micron. They can be spaced a few micrometers, and when you make the calculation, and even on the back on an envelope also, you turn out that on the size of a single microscope slide, you can fit enough DNA colonies or DNA clusters in such a way that you can simultaneously sequence the equivalent of one human genome. It's a simple calculation. And so from the beginning, and thanks also to things all my background in this, as I was working on DNA sequencing technologies starting during my PhD in Strasbourg and later in Ottawa and Bordeaux, I really realized that once you are at that scale, and given the way that you can uncouple the biochemistry and the image acquisition, this gives you the opportunity to sequence the millions of nucleotides a second eventually, and millions of different sequences simultaneously. And when you just make the calculation, which is not a big thing to do, you realize the number tells you that's a genome a day, basically. And so you can imagine the costs also very low, a few hundred euros. That was predicted by making a simple calculation. And so you quickly realize that you can do that whole population genetics, which was needed to find the roots of disease, but also to make hypothesis-free sequencing. By that time, when you wanted to sequence a particular fragment of DNA, you needed to have an idea of what you want to sequence. With the approach also, as I was thinking of it, you could take the DNA, cut it in pieces randomly eventually, and then get each piece on the surface, get a DNA colony, and sequence it. And so you don't, that means you can have hypothesis-free applications like the one that I use today, also not only for the genetics of disease, but to find the organisms that are in a given environment. For instance, what has been done to discover the presence of coronavirus in the first patients of COVID patients, or for forensics, you can now find out who was in a room just by the DNA that's floating around, heard recently, or you can find the microorganisms that are involved in some diseases. People used to think they are purely inflammatory and complex, genetic linked, and it's controversial, but some of them are. For Alzheimer's, for instance, there have been some interesting data for multiple sclerosis that microorganisms could be involved in such diseases. So it opens not only the population-wide sequencing, but also what I call the hidden biology that you can follow up without seeing the organisms themselves.
Okay, great. You mentioned Ottawa, and the audience may not know, but you actually did a postdoc at the University of Ottawa, so there's a Canadian connection to your work. How important was that period in your career?
Very important on both professional and personal way. Professionally, I was at Gary Slater's lab, who by that time was a DNA electrophoresis theoretical guru, who really everybody knew him and was very keen to work with him. He learned me to make simulations, theoretical calculations that allowed me to realize that the DNA colonies would indeed be forming, that they would be that massive, etc. And also Guy Drouin, who was very keen of meeting, because he was working on something where he was attaching with a DNA polymerase a single nucleotide at the end of DNA fragments, which I thought was a great thing for other reasons, and that later allowed me, while I was thinking about the massive parallel sequencing, to realize that eventually you would be able to sequence base by base on these DNA colonies. And so these two things were very important to me, to my family also, because Gary, his wife and daughters, and members of the teams like Ghislaine and Mario, they made us a very good time in Canada, and we were very close to settle there. But there was some immigration incertitude that made us go back to France. But we loved it there, and we were very close to eventually become Canadian, because it's a complex process.
Okay, so thanks, Pascal. I'd like to bring the three. I've got Michelle as well, but Michelle, you'll have to hold for a minute, because I've got a question from the audience to the three DNA sequences. Michelle, you can answer it too if you like. But the question, which you perhaps might imagine, is from Artem Babian, who's one of our early career investigators actually at Gairdner. And he asked, do you think there will be another multi-log drop of cost for DNA sequencing with further generations of technologies? Who wants to take that?
Happy to have a go. Let's go on there. I think we've seen a sort of 6 log drop over the last 15 years. And I don't think we're going to see another 6 log drops anytime soon. But actually, the driving force now is, so the cost of a human genome is literally a couple of $100. And it's reached the stage where the cost of preparing your DNA sample, or the cost of analyzing and interpreting the data, is becoming the bottleneck. So the cost of sequencing could be 0, but you still have these other costs. So I actually think the challenges that will make a difference are perhaps in these other areas, particularly in analyzing and interpreting large data sets to extract actionable information. And so my suggestion would be that's an area to push hard rather than lowering the cost of the middle bit.
Yeah. Pascal, do you agree?
Yeah, yes, I agree. The costs they could still go a little bit down, but what Shankar said is true. At some point, it's only a part of a total cost, and I think it's the simplification of sequencing that could be of use also for maybe for diagnostics applications or also for organism identification. Imagine you are gathering some mushrooms in the forest, and you would want to know, can I eat it or not? You could have a small sequencer that would allow you to identify really the species or other botanicals that will soon make certainly the biggest part of our food. So I quite agree on this, and the data analysis, yeah, exactly, and is some important point. I see it now as I'm also working on artificial intelligence for data analysis and other things. So yeah, I agree with Shankar, basically.
Okay, great. Thanks very much to all of you. Now I'd like Michelle back on screen. We're going to switch from DNA sequencing to CAR-T therapy. Michelle. So unfortunately, Dr. Escher from Israel is unwell and unable to join us. So Michelle, you get to answer the questions on CAR-T therapy. And in particular, I just wanted you to give a little bit more background on the basic science, essentially, again, that between the two of you, Dr. Escher and yourself, to develop the effective chimeric antigen receptor approach, which is really the underlying science behind what's now, current clinical treatments and successful clinical treatments. So can you give us a little bit of background there?
With pleasure, Janet. First, I want to thank the Gairdner International Award for this recognition and congratulate also the other awardees. To address your question, I have to take you back to the late 80s. It takes time to advance therapies. And this one, you can trace back to work of Zelig and my own to the late 80s. Zelig is a basic scientist and immunologist, and he was interested in how T cells recognize the antigens. Antigens is the name that immunologists use to characterize the molecules that our immune system can identify as friend or foe. And if they are foe, they need to act. Zelig created a new fusion receptor by introducing elements of an antibody into components of the receptor that is naturally used by T cells. He did that as a basic science exercise. In parallel but independently, in the late 80s, I formed the idea that we should perhaps instruct the immune system to, in particular, attack cancer cells. We knew at the time that T cells on occasion could do that, but most of the time they failed, which is why cancer is so successful in defeating the immune system in most patients. To do that, we would first have to know how to provide that instruction to a T cell, which would have to come in the form of a genetic instruction. And that meant, how do we introduce a gene in a T cell? So I went to MIT to study what was the very beginning of genetic engineering. And once I mastered, if you like, that nascent science, I went to New York and started to test different receptors, including Zelig's, which I could now introduce not in what are called model systems, but in real T cells that we can collect from anyone's blood. I actually found that Zelig's molecule could redirect the specificity of T cells, but it could not sustain those T cells. Those T cells would die soon after. So throughout the 90s, my lab studied how to increase the way these receptors might work in T cells. We call that signaling. And the important part is that we studied signaling in real T cells collected from peripheral blood. And I think that was very unusual at the time and very critical in providing us the right guidance to molecules that ultimately could be used in the clinic. And we identified a target that's called CD19, which we thought could serve as the first target for clinical trials. It is a molecule found in a number of leukemias and lymphomas, modeled that in mice. And then we faced the issue, how do we bring this to a clinical trial? The pharmaceutical industry didn't develop at the time any cell as a medicine. So we had to do it ourselves. With my colleague Isabelle Rivière at St. Caring, we established a facility. Others did at some other centers, and altogether, these 3 centers initially, in the early 2011, 12, 13, came up with really astounding results in patients who had relapsed refractory disease, meaning patients for whom there is no longer any known option. A single infusion of these T cells that were genetically engineered with a receptor that Zelig initially called a T-body and that I renamed a CAR, or a chimeric antigen receptor, we could induce these formidable responses, many of which have proven to be curative. And this we refer to as a living drug, a CAR T cell.
As a transformational therapy, and with a lot more opportunities and new diseases in the future that this will be applied to. I do want to just quickly point out that you have a very close connection to Canada as well. You're actually a joint Canadian and French citizen. And you did your PhD in Edmonton with one of my early mentors, Tom Wegman. So obviously the Canadian experience has had an effect on your career and your life.
It definitely did. My mother and her family are from Canada, but she had settled in France, where I was born and obtained my medical degree. But while in Paris, I had met this scientist, Tom Wegman, who was visiting from Edmonton, and he told me about how wonderful the University of Alberta was, and so he invited me there, and I welcomed that opportunity to work with him, work with a fantastic group in Edmonton, with Baggy Singh, Douglas Green, and others, and stay with my family for a few years. We owe a lot to the mentors that teach us. In medicine, I had a wonderful professor of medicine, Gabriel Richet, who really shared this passion for physiology and understanding disease. With Tom Wegman, I learned not only immunology, but also to not be afraid of unconventional ideas. And I see you smiling. I think you probably recognize Tom Wegman there. And then at MIT with Richard Mulligan, I really learned genetic engineering. And so I think I owe a lot to these mentors, and I owe a lot to my time in Edmonton. In fact, in some ways, I feel like the idea of genetic engineering of the T cell responses, for me at least, was born in Canada, born in Edmonton.
Great. Good, thank you. At this point, we have a student question, so I'd like to invite all of the international awardees back to the screen. And I'm going to play the video from Popmin, and I am going to ask for very short responses to this question because we're starting to great conversations, but we're going to run through time. So let's run the video.
Hi, I'm Carmen, a first-year life sciences student. Next year, I'll be double majoring in cell and molecular biology and global health. First off, congratulations to you all on such an amazing accomplishment. Now for my question. If you were to pick a single moment in your scientific careers that has been your absolute favorite, what would it be and why?
Okay, so short answers. I've got 4 people on the screen, and I'm going to go in order. Dave.
So I think when I saw a child who'd been a young baby who'd been sequenced using our technology, and both parents had been sequenced using the technology, they'd identified what was wrong with the baby, and she had a drug that cured her, and she was now safely home with her parents. I thought, wow, we've really done something that's made a difference.
Fantastic. Pascal.
Well, not prepared to the question. So one thing that was a marking event for me was to assist to Jean-Marie Lehne's Nobel Prize presentation in Strasbourg, where I was a student. And I was one of the very few students actually at this event. I have many more older people, so I could say, but that was a very inspiring moment. And I think, I don't know, I was inspiring.
Shankar.
I think for me, it's actually I've mentored many students, PhD students, and postdocs, and great moment for me was seeing people that I had the pleasure of mentoring become successful in their own right, scientifically.
Great. I think that's something that we all enjoy. Michelle.
Well, it's perhaps at this moment that I understood that I understand better what I was doing. I was just I formulated an idea. I went to schools where I could learn what I needed to do to implement the idea. I found a university where I could develop my own research. There weren't many people who believed it. It was said that it wouldn't work. Worse than that, it was said that even if it worked, it probably could not be used. But I stuck to it. And today it's just wonderful to see hundreds of laboratories, regulatory agencies, patients, all asking for more of something that not so long ago was just deemed to be futile. So I just reflect on the people who said, well, this could never work. I think of Claude Bernard here, who said, French physiologist, who said, it is what we think we know that sometimes stops us from learning more.
So keep faith. I think all of you have kept faith with your discoveries and seen them carry through to fruition. So congratulations to all of you. Thank you very much. We're now going to move on to the rest of our laureates. And now I'm going to move on and bring to the stage our two Canada Gairdner Momentum Awardees, Dr. Megan Azad and Dr. Christian Landry. And so when they join me on the stage, we'll ask their questions. There we go. Great. Congratulations to both of you and each other. So I'm going to start with you, Megan. And your research program on breast milk and its impact on the microbiome and infant development is fairly collaborative. We saw that in the video. It's international. How do you make those collaborations work? What are the key components to making an international collaboration successful?
Yeah, it truly is a super collaborative type of research that I do. And that's fundamental to all the findings we have. And I think what makes it work, probably two things. One is more the logistics side of it. So, platforms and technology and agreements that we develop to get everyone on the same page. Because when you're working across cultures and countries, even within the same country and across institutions, that communication and clarity is really important. But I think the more sort of exciting part of it to me is the people. So it's about finding the right people to join your team and to work with, people who buy into that collaborative spirit. So who also believe in the synergy of bringing people together from different disciplines who can achieve so much more together than any of them could alone. For example, my own background is in biochemistry and then epidemiology, but I have data scientists who do artificial intelligence and machine learning, and I have public health nurses who work with me, and I have nutritionists and microbiologists and people who know things that I just don't. And so we all need to work together to have this work. And so it's about finding people who believe in that team spirit as well.
Great, congratulations. And I admire the way you manage those things. It's incredible. I know, as sort of another Gairdner connection here, I know you've been inspired by one of our Gairdner Global Health Award laureates, Dr. Cesar Vitora, who I think we both love.
Yes.
Can you give the story of how you met him?
Yeah, it's such a great story. And it really came back to me this morning when people started sending me congratulations messages about that article in the Globe and Mail, where there's a profile of each of us and a little cartoon head. Because I vividly remember eating breakfast one morning, having my oatmeal, sitting on the couch, reading through the Gairdner Globe and Mail announcement of the winners. And Cesar Vitora was the very last one listed. And I'm, reading about the person who discovered NMR and developed a vaccine for meningitis, and then Caesar Victoria for his work on breastfeeding. And I just like spit out my oatmeal, and I thought, this is amazing that breastfeeding is being recognized on this scale, because I think it's been such an understudied and underfunded area of research, and to see that field that I was just new in at the time being recognized on that scale, it was a time when I was feeling, a little uncertain about what I was doing, and that just gave me so much confidence. And I wrote to the Gairdner Foundation, like I didn't know anybody, but I just sent a cold email and said, hey, is there a way I can meet this guy? Like, is there going to be a ceremony? And they said, sure, it's open to the public if you come to Toronto. So I just booked myself a ticket and went to the ceremonies, and I got to meet Caesar Victoria and see his award lecture. And it was so inspiring, because just like we heard from one of the awardees on the screen, he was doing something that was very different at the time. It was an era where infant formula was touted as this great scientific innovation, and breastfeeding was passé. And he was one of the first people to show, actually, there are really important benefits to breastfeeding and risks to formula. And he was very brave, I think, to publish that, and people weren't necessarily buying in. So that was really inspiring. And then even more than just meeting him, which was my goal and was really exciting, I met others at the mingling events around that talk and event. And it was some of them who were arranging a session at a nutrition conference around infant nutrition. They invited me to speak, which I did. And members of the Gates Foundation happened to be in the audience, sort of scouting on that day. And they came up to me afterwards and said, we really enjoyed your presentation. Could you share your slides? And I found out later they used those slides to pitch to the foundation that they should invest in breastfeeding research, and they should ask me to lead the project. So that's what led to my $5 million grant to establish an international consortium on breastfeeding, and kind of eventually led to this award. So it's amazing to me, 7 years later, to be here as an awardee and seeing myself in the Globe and Mail. Just so exciting.
Well, I must say, from the Gairdner point of view, it's exactly what we hope to see. It's wonderful to give people awards, but the point is to really use the awards and leverage the awards to spread more interactions, more networking, bring more people, and build science around the world and in Canada. So it's a great circular story. So
absolutely.
Again, so I'm now going to ask Christian. So Christian, your work is really basic science, but like all basic science, it has implications for human health. So your work is on cellular networks, and it reminds us, as though we must know, genes don't work in isolation. A single gene mutation in a cell is going to have all sorts of consequences on cellular behavior, on cellular networks. Can you give us an example? I think you gave a little one in the video of how this understanding has implications for understanding disease and therapies.
Yeah, thank you, Janet, for the question. And I want to congratulate other awardees. And I want to say it's such an honor to receive this award. And to talk about applications to human genetics. So it's true that everything we do is very fundamental. We use model systems to do experiments and do our research. And there's one question that we were interested in, and that applies to human genetics, is why do some mutations cause troubles in the cells and other mutations don't cause any problems? And one of the reasons why this is the case actually comes from evolution. All organisms have sometimes additional copies of genes that have appeared back in time in our ancestors. And these gene copies, sometimes they work as backups. So if you remove one, the other is masking the effect of removal we just performed. But that doesn't work all the time. Sometimes having a gene backup doesn't help at all. So one thing we wanted to do a few years back was to go in the cell and study molecular interactions among proteins. And then we ask, what happens in the cell when we do these experiments, when we remove one gene? And is there something special happening when we do these experiments and the backup copy cannot do its work? And this is where we found that there are some rules that define when a backup copy works or doesn't. And one example is because sometimes these two backups, or these two copies, they have evolved to work with each other rather than masking each other. And if they work together, then if you remove one, then the other copy has no function. It cannot back up the loss of the first one. So this has implication because we know in human diseases, mutations that cause disease
Great. So I just sort of reading your background, I know that you've been quite heavily involved as a strong proponent of open science and open publishing. And I guess quick comment from you on whether you feel, particularly perhaps in the publishing scene, where rapid change is occurring, is it changing in a meaningful way? What else needs to happen to make everything a much more open?
Yeah, just quickly. So things have changed so much since I've been in science. I mean, I published my first paper, I think, in 2001. And at the time, what you needed to provide was the minimal information required for people to trust your work. That was basically what was needed. And the second thing that happened at the time is that people needed to pay to read your paper. Now the tradition, at least the convention in my field, in genomics, is that you have to provide everything that you've done with the paper, including all the raw data, the code for the analysis, so that people can replicate it. The other thing that happened also is that most journals now, or many journals, offer open access publications, which means that the authors pay the journal, but then everyone can read the paper, which makes sense when you think that taxpayers are paying for what we do, so they should have access. So what remains to, and the other thing that changed a lot is that people now publish their papers before they are published in journal. They publish them as preprints, and those preprints are available all the time to anyone in the world. So things have changed tremendously. There are still some challenges, like the open access venue, for instance, in journals, cost money to researchers. So granting agencies have not, they've not changed the way they fund research to make that feasible in countries like Canada, the US, or in Europe. But that's even more of a challenge in countries where there's not enough funding to actually publish in these open access journals. So we still have to find a way to make this system, which is much better because it's accessible to everyone who wants to read the papers, but we have to make it accessible so everyone in the world can publish open access papers. And I think there are some ways to do that, and some journals from scientific societies, for instance, they waive the publication fees for some authors who cannot afford it. So I think there are many ways we can still improve the system, the publishing system, but we have done a lot of progress in the past 20 years, and I think it's going to be very different in the years to come as well.
Thank you. That's great. So thanks both of you, Dr. Azad and Dr. Landry. And now I'm going to bring our last laureate to the stage, and that's Dr. Gagandeep Kang, and Cherry Kang, and she's going to join us. And so thank you. Lovely. Congratulations to you, Cherry, on this great award. And, you have really focused your work, as you said in the video, on in India on enteric diseases. You set up one of the largest single birth cohort studies studying rotaviral infections in the world. And so from that study, what were the major findings, and how did it impact on actually the ability and the introduction of rotavirus in vaccinations?
Well, the cohort study was one big part of what we did, but we also had accompanying studies. So if I take them all together, I'd say we really did four things. One was we were able to show to the government of India that rotavirus was the major cause of gastroenteritis in children. It was about double of what had been estimated before. So that burden estimation was important for policymakers. The second thing we did in the cohort study was actually to try and look for a correlate of protection. We failed at that, but the data that we had showed us that despite the hope around rotavirus vaccines, the likelihood was that they were going to behave like polio because children were in a poor environment. The vaccines probably would not work as well as we hoped they would. We also evaluated rotavirus vaccines in the laboratory and in the clinic, and that's what resulted in the two vaccines we have. The fourth thing we did was measure the effectiveness of vaccines, and we showed that our predictions were actually correct, that rotavirus vaccines have an effectiveness of 90% in high-income countries and about 50% in developing countries. So they remove a lot of the burden exactly where it's needed, but not all of it.
So that sets you a challenge for the future to bring that effectiveness up. So you have now, having all this wonderful work in India, I know you're still very much involved with research there, but you now have a position at the Gates Foundation. And can you tell us what's the goal in your new role at the Gates?
Well, one of the major reasons for coming to the Gates Foundation was precisely to solve the remaining rotavirus problem. What is it that we can do to improve the performance of oral rotavirus vaccines in the developing world? And perhaps learn from that what we could do to improve the performance of all oral vaccines. It's very ambitious, it's very difficult to do. It will bring together every player that we can think of in the research world and the global health world. But I think this is the right place to be bringing science to global health problems.
Well, that's fantastic, and we all wish you the very best of luck, and I know that you're going to drive this extremely hard with a lot of support, and again, bringing everybody together to address this problem. Fantastic. I'm going to bring back Megan and Christian to the stage for one last student question. It's a question that usually gets asked by students, so we'll see what your answers are. Can we run the video, please?
Hello, I'm Adeline, and I'm currently leveraging AI to reduce diagnostic times for women's health conditions like endometriosis. My question is, what advice do you have for aspiring scientists like myself?
So, some quick words on advice to the aspiring scientist. Megan.
I think my biggest advice is to not be afraid to change directions. I've done that several times in my career, and it can feel like a big risk when you do it. But if you're not feeling the passion or the people you're with, you can change. And also to take some risks. When I went to meet Cesar Victoria, I felt pretty nervous about it. I wasn't sure if it was really a good decision, and then look what it led to. So I think when you're early on, try different things. Don't be afraid to change and take some risks.
Christian?
Yeah, I completely agree with this advice. I think we should, as scientists, we should never feel comfortable. If we feel comfortable, it's because we already know what we're doing. We should try to move around, try to learn new things, and feel that we're always at the verge of not understanding what's happening. And that's where I think we're more motivated and that we can make more contributions. So just explore as many things as possible. That's what I would say.
Cherry?
I think this is the most exciting time to think about a career in science because you have so many tools available to you. Adeline's using AI, and that's, she's trying to use the best of what's available. So my advice is, one, don't be afraid to ask for help. There are lots of people that know lots of things that would be very willing to help. And the second one is persist.
Persist. I think if we had brought back everybody from the other awardees, they would say similar things, and we heard their messages. We heard that they're working in the lab on something that really is not obvious it's going to have the major impact the next-gen sequencing have. You heard from Michelle that nobody believed that CAR T therapy was going to work, but he just stuck with it. So sticking with it, not being afraid, taking risks. And really following your passion, I think, is really a message that's come out of all the discussions we've had today. So I would like to thank everybody, all of the laureates, congratulate them all again, and wish everybody a great day ahead. Now that I'm sure you're all getting emails, I hope, from around the world congratulating you on awards. I'd like to hope the audience enjoyed our discussions. I certainly did. This whole presentation has been recorded, and it will be available if you want to watch it again on our YouTube page at Gairdner Awards in a few days. We also appreciate your feedback. Thank you for the audience. Tell us how you enjoyed the experience. You can fill out the survey. It'll be in the chat, but it's also, we'll send it to you by email. And please follow what's happening with Gairdner at our website and through our social media, and join us live to meet the laureates in person in Toronto for Gairdner Science Week in October. So congratulations again to everybody. Thank you for joining us today.